Hi class, in this recording we're going to start working through some Punnett squares and creating some genotypic and phenotypic ratio examples. So let's look at mom and dad. Mom and dad in this scenario are going to both be heterozygotes. So mom is a heterozygote and dad is a heterozygote. And the trait at hand is dimples. So as we look at dad, I'm going to say that I'll use blue for dad here. Dad could pass on a dominant dimple allele or a recessive dimple allele. And mom could pass on a dominant dimple or a recessive allele. And just for frame of reference, having a dimple is a dominant trait. So if mom and dad reproduce, mom's offspring may have a 50% chance of inheriting her dominant allele. They also have a 50% chance of inheriting her recessive allele. And the same can be said of dad. His offspring have a 50% chance of inheriting his dominant allele and a 50% chance of inheriting his recessive allele. So as we look at these genotypic ratios, we have homozygous dominant, we have heterozygous, and we have homozygous recessive. So from a genotypic ratio perspective, we have a 1, capital D, 1 homozygous dominant. We have 2 heterozygous, and it doesn't matter if you go which you list first, but by convention, we typically, when we're working with heterozygotes, are going to list the capital or dominant allele first, and that's just convention. Although if you went with a lowercase d and then a capital D for the heterozygote, that's all right too. And then finally, we have 1 homozygous recessive. So we have a 1-to-1 genotypic ratio between homozygous dominant, heterozygous, and homozygous recessive. And in terms of the phenotypic ratios, the trait being expressed, we have 1, 2, 3. Let me check this off here. We have 1, 2, 3 of the potential variations where there's a dominant allele. So we'd say 3 dimpled, and then there's only 1 potential variation where there's no dimples. So 1 smooth cheek. So we'd have a 3-to-1 ratio, dimpled to non-dimpled, or dimpled to smooth-cheeked in these individuals. Now that was just a simple dominant and recessive trait. We can also have a multiple allele trait, and with these multiple allele traits, we're going to have more than 2 alleles, more than a dominant and recessive allele when we're looking at a specific gene. A lot of times we're going to have codominance, where we can have 2 dominant alleles be expressed at the same time over an individual recessive allele. The classic example of this is the ABO blood typing, usually abbreviated as I, capital A, capital I, capital A, capital I, capital B, and then a lowercase I. A lot of individuals, though, just like to go A, B, and O when writing out the genotype. And I'll admit, I tend to be more of that end of the spectrum. So let's look at mom. Mom's blood type is O. O is the absence of the A or B genotype. So we know that mom's genetics are OO. She's homozygous recessive for O. Dad is a heterozygote for type B. Now he's type B, not type AB, not type A. So because he's type B, we know he has a dominant B allele. And because he's a heterozygote, we know he has something other than B. Now, if that something other than B was an A, his blood type would have to be AB. However, he's only type B, which means he has a dominant allele that's masking a recessive O allele. So as we look at mom, mom is going to have 2 recessive O alleles. Dad is going to have a dominant B and a recessive O. And as we work through this Punnett square, we find that half of the potential offspring are going to be a heterozygote for B, and half of the potential offspring are going to be homozygote for O. So for our genotypic ratio, we'll have 2 BO paired with 2 homozygous recessive, or 2 OOs. And then for the phenotypic ratios, we'd have 2 B blood types paired with 2 O blood types. So we'd have a 2-to-2 phenotypic and a 2-to-2 genotypic ratio in this instance. As we look at codominance, this is when those traits are going to be equally expressed. And just to review again, blood typing is a multi-allele example of codominance. It's the classic example. So if somebody has a dominant A and a dominant B allele inherited for their genotype, their phenotype would be type AB blood. So let's do another blood typing one. Let's look at mom here. Mom, oops, give me a moment. Mom's going to be a heterozygote for A. So because she's a heterozygote for just A, she's going to be, her genotype is AO. Dad is a heterozygote for type B. So he is going to be a BO. So as we look at dad's alleles and mom's alleles, we plot them out in the Punnett square, and then we go, okay, mom could either pass on an A or mom could pass on an O. Dad could pass on a B, or dad could pass on an O. And this gets to be a little bit more complicated. As we look at this, we have, for offspring, one of the potential offspring is a heterozygote for AB. And then we also have heterozygotes for A, heterozygote for B, and then homozygous recessive for O. So the genotypic ratio here is going to be 1 AB, I can hardly write with my computer mouse, so I'm just going to verbally tell you. We'd have 1 AB, 1 AO, 1 BO, and then 1 OO for the genotypic ratio, a 1-to-1 to 1-to-1 genotypic ratio. The phenotypic ratio is going to be 1 for type AB, 1 for type A, 1 for type B, and then 1 for type O. So there's 4 genotypes and 4 phenotypes when we have this particular mom and dad reproduce. We also can have incomplete dominance. Incomplete dominance is when we have traits blending together. And these traits that blend together have just an intermediate. And the classic example in most biology classes is a red flower and a white flower breeding together to make a pink flower. But this is human A and P, so I want to go with at least an animal example for you. But finding a single allele example of a blending or intermediate dominance with an animal is kind of hard. I could find one with horses though. And this is going to be the horse colors. It has a nice, wonderful phenotype that you can see. We can have brown, we can have white, or we can have the tan in between. An example included in your textbook for a human condition is whether or not somebody has too much cholesterol, hypercholesterolemia. However, looking at pictures of people that do or do not have hypercholesterolemia are kind of boring. You can't see somebody's blood cholesterol content by looking at a picture of their face. So I decided to go with the horses for our presentation, just to make a point. So, mom is a chestnut horse. Let me back up a slide here. Mom is chestnut, so she's going to be the dark brown. And then dad is a palomino. He's a light brown. So as we go forward here, mom is going to, I'll give mom a capital B. She has 2 dominant alleles. Dad's a palomino, so he's the light brown. So he gets a capital B and a lowercase b because he has a mix of dominant and recessive blending together. And as we look at the children, the potential offspring can have that dark coloration, be homozygous dominant, so they'd be chestnut, or the potential offspring could have a dominant and recessive allele blending together to be a palomino. So we'd have a 2 homozygous dominant for the chestnut individuals. And then as we look at the palomino, I'm sure I'm pronounced palomino individuals, those horses have 2 potential chances of happening. So we have a 2-to-2 homozygous dominant to heterozygous genotypic ratio. And for the phenotypic ratio, it's 2, I'll say C for chestnut, to 2 P for Palemo. And when we're working with this 2-to-2 ratio, a lot of times students will ask me, hey, can I just say 1 BB to 1 BB and simplify out the ratios? Mathematically, yes, but by convention, when working with Punnett squares, it's usually best to not simplify the ratios so that we can always know that it adds up to 4. So as a rule of thumb, we want 1, 1, 1, 1, or 1, 3, or 3, 1, or 2, 1, 1. We always want to make sure that these ratios add up to 4 so that we can account for the 4 potential possibilities of our 1 gene Punnett squares. That's all we have for this recording. If you have any questions or comments, please feel free to post them in the class discussion board or to shoot me an email. And as always, happy studies.